On behalf of SciArt Services, welcome to one and all of you gathered here. A very warm welcome to Dr. Deepika Rajesh and to all the participants and to Shruti Govindrajan, who's going to be the moderator for this session. Just a few quick words about Deepika. Dr. Deepika Rajesh is a senior director at Fuji Film Cellular Dynamic International, where she heads the R&D division of the Discovery Research Arm and oversees the production of stem cell-derived products for research use in cell therapy applications. She's a cancer immunologist and stem cell biologist with nearly 25 years of experience. She has a PhD in applied biology from Cancer Research Institute. And she also has over 25 patents. And she has a lot of hope in stem cells for a lot of therapeutic applications for cancer, autoimmune, and rare diseases. Deepika is also very passionate about getting kids interested in STEM education, and she plays a very active role in the Capital Science Engineering Fair, and she's very interested in getting a lot of minorities to participate in it. So please do reach out to her if you have any questions. She also works, she also does a lot of volunteering work for the Madison Opera, for Wisconsin Youth Symphony, and the Milhamni Foundation. She is also a very nice, human being. That's what I would say. I mean, I'm trying to think of the best way to describe her. She's extremely compassionate, and if anybody has a need, she'll be the first person to be there, regardless of whether she knows them or she doesn't know them. And they say that the measure of a person's character is about what they can do for somebody who can absolutely do nothing in return for them. And Deepika clearly meets that. She's one of those people who will go out of the way to do things for people who will not, probably ever encounter her in life. And that to me is the true definition of a human being, a good human being. So we're very, very privileged to know her and have her in this community. She's a very silent achiever. I didn't know for a long time that, despite being a very close friend, I didn't know that she was a director of R&D in a company. That's how quiet she's about her achievements. Yet she's very passionate about getting other people to, succeed. And, that is again another true measure of a human being who can help other people succeed. And she's clearly doing a lot of this with kids. She wants them to succeed with STEM areas. And we thought she would make an excellent fit for this forum. The other thing that Deepika also does is she's very passionate about music and is a very good vocalist herself. And again, that's another thing she's very, very quiet about her achievements. So very warm welcome to you, Deepika. Such a pleasure to have you here today. We have the session being moderated by Shruti Govindrajan. She's a rising freshman at Stevenson High School, Lincolnshire, Illinois. Shruti is passionate about integrating science and arts to enhance the lives of neurodiverse populations. She teaches music and she works with neurodivergent kids and serves as a student ambassador for teen mental health. Very warm welcome, Shruti. Good to see you in this role. I've always seen you as a Carnatic vocalist. Welcome again to one and all of you. We'll start without further ado.
I'm just blown away by this introduction, Vanita. I've known her for almost 25 years, and she's such a wonderful friend, and she has touched so many hearts and almost all of us. She's such an important part of our community. I am so grateful to her in many, many ways to bring back music in my life and a very solid and good, long-lasting friendship, and I absolutely cherish that. She's a very brilliant engineer, a musician, and has started this great organization. So I would like to really thank both Vanita and Suresh for giving me this opportunity to connect with the kids. And I would like to also thank Shruti for being my partner during this talk. So thank you very much once again, Vanita.
Thank you so much for this wonderful opportunity. I'm so blessed to be here.
Just a quick note. I would request whoever can please turn on your videos so we can make this a nice audience for our speaker here who's taking the time out of her busy schedule to be here. So if you could all turn on your videos, I would really appreciate it.
So thank you all once again. And I'm going to let Shruti be the driver of this conversation. But before we start that, and I think Vanita has already talked about my journey, but I just had a one slide summary of what I did to be where I am today. So just bear with me. So if you can see the PowerPoint, and so this is just the one slide summary of where I am. So I was born in Nagpur, and I think that brings us the connection back to Lakshmi, because it's amazing to have people in Madison who are still rooted in Nagpur. So I was born in Nagpur, did my initial schooling there. Most of my education was done in Bombay, which is now Mumbai. A part of it was done in Baroda. And I think the longest that I have stayed in any place in my whole life is now Wisconsin, Madison. In terms of education, I did my undergrad in biochemistry, a master's in microbiology, and my PhD was in the field of applied sciences, which focused mainly on cancer vaccines. My areas of interest. All along, I did not know that I'm going to be here, but there were always certain things that attracted me more, that made me very inquisitive. So, something about the immune system, as to how are we able to ward off so many infections all the time? Why are we not able to control cancer? Why does the immune system become impaired? And finally, the great power of regenerative medicine. We think of regeneration as like regeneration of the limb when a lizard's limb is cut off. And we also wonder as to why did we lose it as we evolved? So those are some things that I was always interested in. In terms of my career, I have spent, after I finished school, I think a good part of it was spent at UW-Madison. And I think now a second part is very soon cellular dynamics. What roles I have done as I have come through this journey is like I have for the longest time being a bench scientist. So I would be like conducting experiments, and which would be like hands-on skills, which is a very essential part of being in this business. You also turn out to be like an author, not knowing that you will still be authoring papers and manuscripts and grants and patents. So you don't do learn to write. And then finally, as you grow into this role, you start off as a solo person, but soon you are training people, soon you're managing teams like any other job. And like Vanita said, volunteering has been a part of my life, whether I was an undergraduate or any stage in life, a parent, a friend, volunteering has been a part of my journey. So that's all I have to say about me. So now I want to let Shruti be the driver for the rest of the conversation. Thank you very much. I'll stop sharing. So.
Yeah, thank you so much. I was just wondering what inspired you to start researching in this particular field other than your interest for it?
So this field didn't exist, Shruti. So it was not something like, oh, I just woke up one day and I said, I want to work in the area of stem cell. This field is so new that it only opened up, at least the kind of research that I am involved in. The beginning of that field happened in 2002. So you realize how young this field is. I would say in the field of biology, the fields are interconnected. So I was still interested in the immune system. And so it became a part of my graduate studies. It continued to understand the immune system from the cancer perspective. And very soon, I think just something happened, like being at the right place at the right time. The biggest breakthrough in the field of stem cells occurred here in University of Wisconsin-Madison, when Dr. James Thompson showed for the very first time that you can actually culture stem cells in a dish. My lab was where I was working, where I was doing my postdoctoral fellowship, was very adjacent. So it was a breakthrough discovery. It was a science paper, and we were all enamored by the field of stem cells. And it was an opportunity I couldn't, let go. And it perked my curiosity, and I grew along with it. So it happened very serendipitously. I didn't get up as a child and said, I want to be a stem cell scientist. So.
Wow, that was very inspiring. I did not know that the breakthrough actually occurred in Wisconsin.
Yep.
It's very cool. Um, yeah, so I learned that you worked with IPS
Mhm.
cells. So what are IPS cells like?
Okay, very good question. And thank you for Googling me and finding out that I work. It's a very good question. And I think to answer that question, I want to just take a step back and tell you a little bit about how many different type of stem cells are there. So out of curiosity, I'm going to ask you, what do you think? When we say stem cells, so you think they are a kind of cells? You think?
We can actually open these questions to the
Yeah,
audience too. There are
sure.
lots of, and anybody can interrupt anytime to ask
Please, this is
questions.
a very informal talk. So interrupt me. I may have answers for your question, I may not, but I think this is how I want it to be, very casual, very informal. So,
Looks like Arjun has a question or an answer.
okay, please.
Stem cells are like the cells that will grow into like your muscle or bone, like those kind of cells. And there's specific types of cells for each like kind of cell. So
Exactly. So they are like a raw material. You call them as a raw material that they have the power to become any kind of cells. But let's take a step back and understand, because this term is used very generically. And what do you think, Arjun? You think we should have buckets and buckets of stem cells, or we should have a few of the stem cells? What do you think in terms of quantity? Just
uh,
what your guess?
maybe like a small layer that like when your normal cells die, they get replaced, and the normal stem cells will get like they'll like regenerate. So
There we have a stem cell biologist in our audience today. So so very good, excellent. And let me just share one textbook picture of I don't know whether I'm still sharing. I am, right? Okay, let me. So very good answer, Arjun. So I just want to give a 2-second introduction of stem cells. There are different flavors of stem cells, and unfortunately, everything is called stem cells. So there are stem cells, like Arjun said very correctly, that there are stem cells throughout our body. They don't come from air. They are something that happens during the time that we develop in our mother's wombs. So they get embedded in our muscle and bone, nervous system. So these are stem cells that are like sitting in various areas of your body. So they are important. We are surviving because of that. Because, like everything, like exactly what Arjun said, we have a constant supply of 200 different cell types in our body, all of them cycling at different times. Just to let you know, the intestinal cells that line our gut recycle every 3 days. The red blood cells go away in 120 days. And then there are cells that of the muscle that regenerate after every 10 to 20 years. Who keeps track of this inventory? When do we know that it's time for making another population of cells? Because we need those cells. If all red blood cells die at the end of 120 days, where is the next set of supply coming from? So that is the biggest role of tissue stem cells. Switching gears to another form of stem cells, and this was the breakthrough discovery that Dr. James Thompson did. These are the most precious stem cells, because these are the stem cells that are formed when you, all of us, are developing as an embryo. So they are a cluster of cells that are at the blastocyst stage. They are like a cluster of cells. So nobody had figured out how to take these cells out and study, because this is a very important tool for developmental biologists. They don't know how to um, they don't know how to. Rajesh is saying nobody can see me. Sorry.
No, nobody can see our presentation.
Oh, really?
Yeah.
You can't see your presentation?
Yeah, you might need to share again. I had to stop your slide share so
Oh,
that your face can be seen more clearly.
okay. Now, sorry. Please tell me about this. Thank you, Rajesh. So the second kind of stem cells are like the embryonic stem cells, as I was saying. They are formed in our bodies when we are growing as embryos. So what Dr. Thompson found was he found a very elegant way to take such an embryo and found conditions where they can be isolated and grown in a dish. People saw these stem cells growing out for the first time outside the human body for the first time, and this was the breakthrough discovery in 1998. So this made a big splash. Suddenly, this was like a tool that was available for developmental biologists, because they could see the earlier stages of how a stem cell becomes like a neuron, a cardiac cell. So they had this powerful tool in their hands. And finally, this kind of stem cells also created all the ethical and religious advertisement that happened, that there were a lot of opposition for working with this kind of stem cells. So the field became smart. So comes 2002, they found a very, very elegant way of making the same kind of stem cells that you see in the inner cell of the blastocyst by taking maybe 2 ml of your blood, a pluck of your hair, or maybe a small piece of your skin. And so this was the power of genetic engineering, where they found that this was around 10 to 12 years of work, also pioneered by Dr. Thompson and Dr. Yamanaka. And here there was a scientist named Janine Yu. So what she did was every gene that was cloned for the human, she began inserting that particular gene in a blood cell and seeing, can we turn the clock? Can we turn the clock? So she found that after doing a tons of permutation and combination, 4 genes were required when entered any cell that had the capacity to replicate. It could turn back the clock. That means if a red blood cell, okay, say a white blood cell is circulating in your body, it has made its final choice. It's going to live like a white blood cell, and a few days later, it's going to just perish. But if you catch hold of that white blood cell and now give 4 genes and give it time, so it'll go back to an early white blood cell. Then it'll go back to a stage where it is thinking of becoming a white blood cell. Then it will go back to a stage where it can become any cell. So this technology was so powerful. And this is the power of induced pluripotent stem cells that, this is exactly the area that I'm working on. And just giving you one more spiel. We should never forget what the power of adult stem cells. So everything that resides in our body, that little schematic tells you that today, all these areas of your body, the stomach, the skin, the liver, the brain, all of us have reservoirs of stem cells. They are very, very small pockets of stem cells. They have come to us before we were born. There are clocks inside these stem cells because they have to persist too. If they go away, the power of regeneration is gone. So they have a certain clock at which they replicate. They also have the power to sense as to what needs to be replenished. The biggest amount of discoveries as to how do you classify, how do you know what's a stem cell is? You give a huge bucket of cells. These cells are not glowing green and blue to say, yay, that's a stem cell. This is a stem cell. So a lot of classic work was done from 1963 to almost, I would say, in the early 70s and 80s. And it was predominantly done in one class of stem cells, which lives in our bone marrow. And why this was important? Because they felt this had the magic bullet for curing cancer. And till this day, this is a very effective way for curing cancer. So just let's talk about one such kind of adult stem cells that has really saved a lot of lives. So it is the liquid that lives inside your bone marrow. And in that, you have stem cells which have the capacity to give rise to everything, all types of cells that circulate in your blood. So understanding those stem cells, understanding what are the signatures, what is it that, how do you know these are stem cells? All of this work came from the work from the bone marrow stem cells. And that's just the ratio. We have like around 100,000 cells of such stem cells in our body, and the total number of cells that are circulating at a time in our body is around 32 trillion cells. Like Arjun said, they are very sparse and very precious and very powerful. And finally, this is the last slide. So what do we do as a business? So coming back to your question now, pluripotent stem cells. I said it's a clever way of making these precious stem cells, which we call our totipotent or pluripotent. That means they can give rise to any cells of your body. What do? We basically take 5 ml of blood that you go for any tests in your lab, you give away some cells. At the company, we have various way to kind of bring back these stem cells to a stage of stem cells. So we have a collection of stem cells. It takes about 3 months. This doesn't happen in 2 days. So this is the time it takes to turn the clock back. Once you have that, we have the ability to scale them up and make like lots of it, and we freeze it. And then from there, based on the demand, if we say that I would like a bunch of cardiac cells, that means cells that beat, they are the components of your heart. I would like a bunch of neurons. Brain is filled with 15, many types of neurons. So there are ways to make excitatory neurons, inhibitory neurons, so class of neurons. Somebody says, I don't care about blood. I just want you to make a synthetic RBC. Like if I poke my hand and I get red blood, go ahead and make this in a dish and show me that in a tube. So all of that is done in the company, and that's like the core business. So did I answer your question, Shruti?
Yeah, thank you. That was a very informative. I'm just gonna go back to the previous slide real quick, and I have a question.
Sure.
Um, how are the stem cells identified in the body, and how are they isolated in order to?
So
Yeah.
so a lot of technical details, so but didn't want to bore you with all that. But yes, so they have a classic marker for on their surface, and many of them, one of them is called CD117 and CD34. So they have certain proteins on their surface that are enriched, which are not there in any of the cells that have decided that they don't want to be stem cells. They want to be something else. So there is a powerful way of like magnetically pulling these cells out from the bone marrow. So it's a long process, and we actually use powerful magnetic beads to actually just select those cells, and they are collected, and those are your precious bone marrow cells. A lot of research, this is just like one way. The other way is that you can open up their DNA and find out that there are very intrinsic signatures that are present to say that this is a bona fide stem cells. And it's very much needed, because without that, you will be just collecting garbage. So you have to make sure that what you have in your hand is a rare, pure population of stem cells.
Thank you.
Yes, Arjun.
Oh, so I have a question. How do you use like how do you use like stem cells to cure like cancer, to treat cancer?
So two ways to answer your question. The newer way of curing cancer is the way by, you must be hearing this term of CAR T immunotherapy. But I'll go back to the same slide, how it was done in the past. What happens in cancer, Arjun? So cancer is something that is not like an infection, like the COVID vaccine, that a COVID virus that has come inside our body and our body is trying hard to fight it. Cancer happens when our own body cells are able to, they acquire some unique, they become Frankenstein cells. A normal sign, they start growing very fast, they acquire some new bells and whistles on their surface. And somehow, since they are a part of our own body, our own immune system is not able to identify them. So initially, there is always this balance, whether how many police guys you have per thief. Very soon, the thief becomes smart, and they have a large army, and they overpower the immune system that we have. And that is what is like, that is why cancer occurs. For the bone marrow cells, what they found out was, okay, the cancer patient is not able to have a strong immune system, and hence they are not able to combat this cancer. So the earlier part of all the cancer treatment went when they would isolate these precious stem cells, exactly what Shruti was asking. And what they would do is infuse it into a patient who is suffering from cancer. So they would have ways where they would give medications and wipe out their immune system. So they know that, their own immune system is kind of impaired. They are not able to respond to the cancer. So they would give a fresh dose of stem cells from another person. It could be a sibling. There is a lot of matching that you have to make sure that these cells are accepted in your body. And so they would infuse that, then with the hope that once they are infused, they know where to go. They have something in their memory to show that they need to go to the marrow. Once they go to the marrow, they sit down and they start multiplying. What happens when they multiply? They suddenly realize, oh, this shelf is empty. There is no red blood cells. Oh, let's make some red blood. Oh, this shelf is empty. There is no white blood cell. So they go and slowly start repopulating healthy cells in that cancer patient. And that cancer patient now gets like a fresh dose of immune cells to combat the cancer. So in a very high level. So
I have a question, Deepika. Is there a direct correlation between stem cell quality and the age of the individual?
yes, there is.
Okay.
Yes.
So how do you
So
ensure that is there a set of criteria that a person has to meet before they donate?
not so on the surface, if you're just looking at the two markers, and all persons may have the same criteria. Where you get the difference is the amount. As you age, the percentage of the stem cells, like from an 80-year-old, the pool that is left in that is going to be smaller compared to something that is derived from a 24-year-old. There is also things that are in all our cells which tell us where we are in our age. So there are aging clocks, like most, there are telomeres that tells you, by there are aging clocks in all our cells. So the stem cells that are coming from an 80-year-old are not going to be that robust. They come in, they are isolated, they exist, they do their function, but when you are now putting them against performance with a 24-year-old versus an 80-year-old, this would have a higher efficiency. The turnover, it will have like better efficiency. It's nothing regarding what you will pull out the same cells. It is those inherent clocks that actually make a difference between one type of stem cells from a younger person to a very nice question. So,
We'll have a separate session on how to extend the telomere length.
okay, we'll do. So I'll take back to Shruti and ask, is that's that answer? And Arjun, of course, are you satisfied with this answer on a high level? Okay.
Yeah, for sure. I just had one question. You briefly mentioned CAR T cell therapy. How is the receptor put inside the patient? How is it given to the patient?
Should we jump to that? Okay, I did have a little slide. These are the last two slides. So let's go back to again cells of the immune system. Before we jump into CAR T, do you know why it's called T and not Q and not P? Why not Z? Why? Why is it called CAR T?
Because of the T cells.
Okay, that's a good answer. Do you think there are other alphabets or other type of cells living in the part of the immune cells? And this diagram just tells you that. So this is our defense mechanism. This is just not like, so that when you say immune cells, we just don't have T cells taking care of us. We have many, many types of cells. So one of the powerful ones are, of course, T cells. And even if you say T cells, there are subtypes of T cells. There are helper T cells, there are cytotoxic T cells. Those are the cells you want, like from the name says, cytotoxic. They can go and kill the tumor cell. They are actually helper cells that run around and get help. Like, okay, come here, there is danger, let's go and, take care of this infection. There is something called as very powerful, called as a memory T cell, because it remembers every pathogen that enters in your body, and it remembers the signature, so that the next time the pathogen comes, it knows that it has the code to kind of attack this pathogen. There are a powerful set of other immune cells, which are also called as B cells. These are the ones that when you get a COVID vaccine and somebody says, go ahead and measure your response antibody titer, they are not made by T cells. They are made by a separate class of immune cells called as B cells. So anytime you have a collection, like you have this plasma infusion, antibody infusion, so that's another class of, the immune system is so elegant and so powerful that we should thank it every single day that we breathe and live, because as we talk, as we breathe, we inhale a lot of viruses, bacteria. And so how are we seamlessly going from day to day? So there is like T cells, B cells, they are sitting, there are also tissue type cells, which are hiding in your spleen, under your arms, your sinuses, that are always sensing, always patrolling to make sure, what is out there, so that you stay healthy, and they are constantly combating anything that comes your way. So this is a generic picture of the immune system. Let's go back to the bone marrow. So as I said, the seed for all the cells that you find in your blood, which is all the immune system, is present mainly in the bone marrow in the type of a stem cell called as the hematopoietic stem cells, which is a part of the adult stem cells. So this is the way, the immune system is evolved. Let's go to CAR T therapy. So what did I tell about cancer? I said cancer is something that our immune system is not able to respond. So people have actually taken cancer cells, and a lot of research has been done mainly with blood type cancers like myeloid leukemia, AML, CML. They have taken these blood cells out from cancer patients. They have really studied hard to say what is so different on their cell surface that the body was refused to see it. Cancer cells are very smart. Even if they have something different, they have acquired something different, right? Because they became a separate pack compared to the normal cells. They also have clever ways to hide it. They develop a lot of hidden moieties so that these new things are never exposed for the immune system. So a lot of these studies came by opening up cancer cells and finding out what is so different about them. And one of such molecules that is currently in therapy is like the CD19. It's a molecule, it's a surface molecule that is seen in most kind of leukemias. So coming back to your question, so we now have the first step of the way. Find out what is different between your normal cell and cancer cell. Yes, we found out that molecule, that's CD19. How do you tell the patient's body now to attack these tumor cells that are bearing that particular molecule, CD19? So by genetic engineering, they go and recreate a receptor. So all T cells, if this is a T cell and this is a receptor, this receptor is the one that looks at a cancer cell and finds out that, oh, there is that molecule that I need to attach. It binds it, and then the killing occurs. This thing is impaired in a cancer cell, in a person who's suffering from cancer. So they take the T cells from the cancer patient, only the T cells, because they are the part of the defense mechanism. And then using genetic engineering, they create every part of the receptor. So it's a lot of molecular biology that goes into play that tells you which is the front end of the molecule, the back end of the molecule, protein engineering, bringing different parts of it together, stitching it together. So now it sits like a receptor. Bacteria are used mainly to make these synthetic proteins. Once you have this protein made and it's purified, all you have to do is bring the T cell from the patient, and by a process of either you give this extra protein to actually a virus, you give it to a virus. So the virus, a harmless virus, enters the cell. It's a mode of delivery. So that then the receptor gets attached to the T cells. There are other ways to include it too. You use going too much into the nitty gritty, but there are lipids that you can add so that the cell membrane gets disturbed and this foreign protein now goes into the T cell. Once you have such a cell, that's not good enough, because if you make one such T cell bearing a small receptor on its surface, that's not going to go and cure the person who is having cancer. You have to have the state of art to then select these T cells and grow them in large reactor. You need billions of cells to come into a cancer patient. So once you select the cell, there is a second part, is huge expansion of those cells in reactors, and then taking it back to the bedside of the patient and then infusing it. So this is a very cumbersome process. Can you realize how many stages there are? Bleed the patient, identify the T cell, find out what is different about the cancer cell, engineer that part which is different on the cancer cell, tag it along to the patient's cells, and then expand the cells and then give it back to the patient. And so many moving parts, incredibly expensive. So you realize it takes around like $200,000 of endeavor to bring such a kind of state-of-art treatment uh, bedside to a patient. So that is the conventional medication that you see called Chimera, Yescarta, all of the ones that made big name. They were just been like 2018 is the time when I think FDA approved. Do you realize how new this field is? And this has just been done with one type of cancer that has CD19 as one of the molecules that they can chase. So the field has just begun. Nobody knows if the same kind of thing can be done for solid tumors. People are always not dying of leukemias. People are dying of uterine cancer, ovarian cancer, squamous cell carcinoma. These are tumors that grow. So will the same kind of methodology work? Will the T cell know that it has to now go inside a tumor and kill? Or should we now think about a different kind of cell, like a macrophage, which is also sitting in our immune bucket and say that, oh, can we now teach the macrophage to carry this payload and then go inside the body and start attacking tumor cells. Solid tumors are actually, to this date, there is no immune therapy for solid tumors. But this is a great beginning. Too much details, but are you with me, Shruti?
Yeah, thank you. That was super, super insightful. I didn't know there were so many steps involved, and I didn't even know it involved protein engineering either. So yeah,
Uh,
definitely didn't.
Biology is it as maybe I would say 50 years ago we were very segmented. A cell biologist would only do cell biologist, a molecular biologist would only do molecular biologist. The fields are merging. So I just feel today a cell biologist will also do a part of protein engineering, will also do a good part of understanding where the genome is and what to engineer next. It is a very, very close field where you also have a lot of chemical engineers coming and telling us what kind of vessels, how should the gas exchange be, how should we grow them, how should we not kill them. And then there is a lot of chemistry that's happening. How do you package them, how do you freeze them, how do you make sure they are frozen at the right temperature, how do you make sure the ice crystals that happen in cells during freezing, they're not, after doing all this, if you now don't know how to freeze them, what is the point? You would never be able to bring them back to life if you didn't know how to freeze cells. So cryopreservation, which involves a lot of physics, a lot of Raman spectroscopy, a lot of chemistry as to how many sugars and salts and what's the molarity, all those things that you hate while studying chemistry in school are very, very much in use at this time to develop like a magical solution that will hold these cells, that will allow these cells to be cryopreserved deep down to minus 196 degrees centigrade. And then as you need, you bring them out. I can segue to how it can look in the future, but if you have any other questions, I would like to pause and ask you if there are any more questions or you want to know anything more about stem cell therapy.
I have a question, Deepika. First
Yes.
of all, this was really, really insightful. And the fact that you talked about these merging disciplines is a very, very important aspect, I think, for students to know, because I think there's also a misconception that you have to very narrowly specialize. And I don't think that's the case. You can also, to get a broad understanding, it's also very, very important, I think. And that's one of the things that, even, for example, now you talked about merging disciplines within biology, but, even across disciplines, knowing math and still being able to do biology, knowing biology while you're doing math, you'll be able to apply some of these basic sciences to a lot of these applications. So that's a very good point you mentioned. I had a question. I've been not been following this discipline much, but so, pardon me if it's a stupid question, but
No,
there was
no.
a time when this whole stem cell bank thing was being majorly marketed, right? Where people
Mhm.
would talk about how, you should go and make sure you save your own stem cells. It'll come in handy later. Is that still, happening? Is that something that people are still very actively, working on?
You said it very nicely when you said it was being marketed well. So,
Yeah,
so I think there was a lot of power in it, and I think at that time we were not talking about pluripotent stem cells. The market that we are talking about is the adult cell market, and the subset of those stem cells are the umbilical cord stem cells. I think there was a big
right. The
push.
umbilical. Yeah,
Each time we would have anybody was going to have a child, they would be
correct.
encouraged to kind of bank their stem cells because what in the future if you have cancer, these will come back handy. um, Without going too much into the details, I would say there are maybe one or two papers that show that it has a shelf life of 20 years. But having worked with stem cells for almost the same amount of time, I have never seen. So we have worked a lot with umbilical cord stem cells because that's our benchmark, right? When we isolate stem cells with pluripotent cells, we have to always benchmark what is so different about the pluripotent stem cells. Are they similar to the cord blood stem cells? Are they similar to the bone marrow stem cells? So we do, for some reason or the other, we seamlessly work with bone marrow stem cells and umbilical cord stem cells too, because we are benchmarking all the time. So at that time, the hands-on experience, let me at least tell you my experience. The hands-on experience for at least 5 to 6 years where we would buy these banked cells and just see, how good they are. I would say the recovery was very, very bad. And I would always feel sad that this has been so over marketed, thinking that you can come back to 20 years later and these cells are going to be just sitting in the right way as they were frozen. So I think even the technology has changed too. So if you had a child 25 years ago, the technology that was used to freeze those stem cells is light years away from how we are freezing stem cells today. So with that poor technology, I said, if anybody had banked their cells 18 years ago, there is nothing living in that vial anymore. So I personally, I haven't banked. I have two children, and I haven't banked
Yeah.
my stem cells. So I think it needs to grow. It has a potential. It will grow. I think we should find better ways to preserve them. More than better ways to preserve them, remember what I said. The cells are always very sparse in your body. There is a lot of engineering opportunities that you have to do to scale them up, to make them amenable for treatment. You can't bring 10 cells in a bag and I'm going to infuse into you and say, wow, they just magically go and cure cancer. No, you need billion cells, 1 billion cells, 100 million cells in a bag to go into somebody to have any kind of efficacy. So even if you froze some cells, how many cells are in the umbilical cord? They are probably 50,000 cells. That's about it at the most, living cells. So you have to now thaw them, have the state of art to grow them to a billion cells. Let me also add one more thought to you. Just by growing them is not enough. They have to stay as stem cells. That is the hardest part because they are not programmed in your body to grow into such large numbers. To stay as the true potential of stem cells, you have to cleverly have conditions so that you're not changing them. Because remember, they are like a cell that can constantly drift to become a heart cell, a brain cell, a hair cell, an eye cell. You have to tell them, like your child, focus, focus. You have to stay as a stem cell. So that's not easy at all. A blood stem cell, if you take out from the bone marrow, 7 days you keep it in a dish, it's no longer a stem cell. It has now decided to become some cell that it wants because it has the spontaneity in it to kind of go any way it wants. So the challenge of having small cells, not having an elegant way to expand them, also preserving the quality of those stem cells are all the reasons why it's not a good idea today to bank your stem cells unless there is like an acute need. If you have a genetically associated disorder that is hitting every sibling and it's very highly prevalent, so if you are in that bucket and you are constantly monitoring your genetics because you have such a very acute disorder, then I would say advocate and say go ahead and bank some of your stem cells. Did I answer your question or gave you
Very,
more
very
questions?
much, Deepika. So one thing I wanted to point out, and sorry, Shruti, if I'm interrupting here. I wanted to also talk about why we're doing this. These conversations are primarily targeting um, kids who want to, be inspired by different disciplines. And in this context, one problem I find, okay, I'm just looking at it from the standpoint of a student. Okay, I'm taking a step back from all this. If somebody wants to be a stem cell biologist, what can they do in their formative years of schooling? What can they do to actually see something hands-on? See, for example, chemistry, you can do experiments at home. Physics, you can do experiments at home. Math, you can do hands-on stuff. I loved biology growing up, but I never took it up because there was no opportunity for me to actually see anything, besides, of course, organisms and things that you can see with the naked eye. There was nothing else that I could do with my hands. Is there anything you would recommend for students who are interested in getting into, say, molecular biology or stem cell biology? What are the venues they can pursue to do some hands-on things or see something that, will inspire them?
First of all, I would say that when you are in school, they do the actual curriculum and understand, like the school is built in such a way that you're getting a sampler. sampling of physics, chemistry, and biology. Don't eliminate those courses and just go advanced in one area, because that's the perfect time for you to see what's there. So definitely take those courses. I'll come to your question, Vanita. I will say for the undergraduate, I would say again, doing a field, and these days of, I think my opinion today, if you ask me, is to take up some undergraduate stream like biomedical engineering. And I like that because it's not skewed. So you get to, like I said, our fields are merging. You need to know to code. You need to know how to understand your genome. You need to have hands-on experience, how to build a tissue. You need to know how to grow these cells. You need to have some kind of engineering background about environment controls. So I think having a major like that is, I think, incredibly valuable, because you are well suited to, again, pick a field. It need not be stem cell biology. It can plug into any types of biology, and these fields merge. um, I think the one of the best things you can do is, like you have for clinicians, I think you need to shadow somebody. You need to shadow somebody in a lab to see what it's you have to have a lot of patience. All these things, that when I say that we made heart cells, we made neurons, just to tell you, to convert a stem cell to one type called astrocyte, it's a 120-day process. So it's patience. You have to learn that every day you say, oh, I just come, I just change some media. I don't see anything. I don't know what's going on. I know these people are making some brain cells. So children are very impatient. It's like not that I had stem cells today, tomorrow I have these beating hearts. It's a long journey. Remember, you are many of these cells are formed in your own body after 9 months of gestation. And we are trying to recapitulate all that in a plastic dish. So also have a understanding as to time. Shadowing is probably the best way to do, but we have limitations now with COVID and other things. It's not very easy. So this is something that I dream of, where I think we have a hub where we set up small hands-on experience. The Wisconsin Science Fair does a very good job where they showcase a lot of things, and so going to many of these fairs helps you to at least see what's in there. Professors are very busy. They have so much on their plate in terms of teaching. So it's not easy for them to just let students come in. You have to find out senior mentors, like who are themselves graduate students or postdocs. They can be the first level of like hands-on experience. And Vanita, more than anything else, YouTube has changed the world. Anything that you need, any technique you need, there are like popular websites that you can go and you can actually watch. One of my favorite journals is actually Journal of Visualized Experiments. So this is a step where there will be articles saying how to make a hair cell from a stem cell. And this person is walking you through, like, I take this, I add this, I centrifuge this, look at under the microscope, this is how it's become. So you get like a most virtual experience of what this person is doing. You don't have to wait for 9 months like how we have to wait sometimes. You do fast forward and they go through many steps. So Journal of Visualized Experiments is a great place for children to see how these things are done. Another great resource, I mean, video is not everything. I think the fundamentals are, you should read. I think the Harvard Stem Cell book is a freely downloadable book where it's a rich collection for people like me who want to know what's happening in the cutting edge, but they have beautiful introductions and schematics to even understand, why are they doing this? Where is this coming from? Why should this be important? So I would say those are all your resources.
Amazing. Thanks, Deepika. This is really, really helpful. And I think for kids who are planning to get into this area, this can be very, very inspirational. Maybe we should do some more talks in the future where you can talk about how, what are the different other areas that people can pursue and what would be great venues to do that. And maybe you could also give us a little introduction about the Capital Science Fair. So we could do a session just on that and how, you can also maybe show successful projects that students have done so we can inspire more kids to join the science fair as well. So Shruti, maybe we can take one last question from you and then wrap up this session because we're nearing the stroke of the hour.
Sure. So I know you were super passionate about stem cells and creating a change in the field. So I was just wondering, where do you see this industry in the near future?
Yes, it's a very powerful question. And there are many, many ways it'll go. I think it has a place in almost everything that we do. It has the biggest application in medicine. And we talked a lot about cancer. So let me say one more thing where it can go so we close that loop. But that's not the end. And I can talk to you about a few other things. So we talked about how cumbersome it is to do this CAR-T therapy coming from a patient. You have to bleed the patient twice and infuse them and all that. The dream for tomorrow is to never touch the patient. So understand different the signatures of different cancer cells, develop those molecules that with protein engineering that you have for different cancer cells. But instead of every time going and bleeding the patient, have one off the shelf product. That means using iPS cells. So what did I tell about iPS cells? It's synthetic, and we already know how to scale them up in billions. So if you have reservoirs of T cells, just T cells made from iPS cells, and they're all sitting. So the second part of it is each of this T cell, one T cell gets like molecule A, molecule B, molecule C. So if you have all these collections of T cells and A can see AML, B can see CML. So each of them are geared for certain cancers and they are just sitting in a freezer. So it would be a breakthrough technology to then tell the patient that, oh, I'm not responding to chemo, I'm not responding to radiation, I think I need my immune therapy. So all you need is to order these cells, the cryopreserved cells, coming to your bedside, and they are infused. So it's really off the shelf. And this is going to be such a big game changer, not even for cancer. It's also going to be a big changer for standard bone marrow therapy. You don't have to ask siblings and matching and all these people donate a part of their bone marrow. So it's going to have a big place in cancer therapy. It's going to be a game changer, and it's not something of the past. There is already a company called Fate Therapeutics who has done their first trial already with iPS derived cells. They are NK cells, they are not T cells, but the same function. Like I said, the immune cell is a big bucket. We only think about T. There are other cell types living in there too. So the NK cells is already in therapy. Our company has partnered with a therapeutic company called Century Therapeutics, which is going to have a second wave of such an iPS-based therapy that's going to be again for cancer. So that's still one bucket. Where is it going otherwise? It's going to go have a big play in, when people receive organs, like cadaveric organs. People die, donate their organs. You are giving a lung, you are giving kidneys. They have a lot of rejection issues because they don't belong to you. So, and these people are put on immunosuppressants for life. So there is a special kind of cells that we can make and again infuse so that these people are not hooked on to immunosuppressants for life. This is and it can be the same thing, like they can someday be a part of a vaccine platform where they are carrying. Why should just they be carrying proteins that are unique on antigens? Why don't we take this a step further and give them proteins that are present on parasites like malaria, something that an HIV protein has? So it is going to really change the way we treat a lot of diseases, but it's not going to happen overnight. The field, remember, the field was just born in 1998, and the way we are trying to work with stem cell now, the first discovery came in 2002. So it is a very, very new field, and it has come so far so quickly, and it has so much potential to grow further.
Awesome. Well, thank
Yeah,
you.
fantastic. Sorry.
Yes, Suresh?
And Deepika, you're such a wonderful teacher. You had me completely engaged on such a difficult topic. You made it a beautiful story. I wish you had taught me biology when I was in high school. I'm going to take that up more seriously. So thanks so much, Deepika, honestly.
I completely agree. I think I would have been totally excited about taking a biology full time if I had spoken to somebody like you. But here is one thing, Deepika. I know we started off saying that this is going to be about career conversations. I would love for you to come and do some lectures on, biology. You know, could be stem cell biology, it could be whatever you choose. but we can try to find an audience for it. It'll be a fantastic thing to do through SciArt Services to, spread awareness on, many of these topics and also get people fundamentally interested in the subject. That would be really nice if we can.
Yeah, the field is like Suresh said. I was actually scared that Suresh is going to now trump me with a very difficult question, but thank you, Suresh, for the kind words. I think the field is very, very broad. Everything that I said is like a small drop in an ocean on its own. So even a simple thing, what Shruti is kept on asking as to what is CAR T therapy? How many infusions does a patient need? Why does it go bad? Why does it not work? Why is it so expensive? So each of this can be sliced and diced and can be a 1 hour conversation. Or going back to the basics, how do you do this engineering? Why is it so hard? And still, at least at a very high level, we can talk about it. No, I thank you for the opportunity, and as you know, I am very passionate about
We're very
bringing
privileged to
this
have
up.
you.
No,
Very
it's
privileged
a
to have
pleasure
you.
of mine, because you gave me an audience, and even if 2 of them take this talk seriously and think about, pursuing this field, I think my job is done. So
I would also like to add over here, this is very phenomenal. It's not just for the kids, it's also for us adults to understand. And, it's pretty much so much work has gone into it, maybe life's work for so many, great engineers and biologists and everything. So thank you for arranging it. And thank you, Dr. Deepika, for
Over the
your time.
years,
Yeah.
thank you,
I've heard.
Nita. Thank you very much. Thank you very
No,
much.
I'm privileged to
Can I
join
also-
this one. And thanks, Shruti and Vanita, too. So thank you.
Can I also add, Vanita?
Yes,
One more thing?
absolutely.
Yeah, thank you, Deepika. It's been a wonderful presentation. And I would like to suggest to Vanita as well as to Deepika, if possible, the high schools, the kids who are in maybe junior high or so, they should hear such a presentation to make a decision for themselves as to where they want to go, if they want to go.
That was exactly the intention of this, Lakshmi. That was exactly the intention. What would be great is if people like you could forward this to your mailing list. And, we have a limited mailing list and presence on Facebook, but this is only going to spread by word of mouth. So the more you can spread this, and I'm going to put out these excerpts. For example, whatever Deepika has explained so beautifully, and we've had Suresh do something before. We've had, another lady called Vanita Kumar who did a presentation on electrical engineering. We're covering different disciplines. Soon, I'm hoping for you to also do that, Lakshmi. You have to talk about your experience here as a scientist and, as a first generation Indian in Madison. Then we also have Dr. Rajesh Rajaraman also will be doing his talk very soon. So we're very lucky to be surrounded by people in this community who are very educated and accomplished. And it'll be nice to draw on their experience and inspire youngsters. So let's, hope we do that. Arjun, I think you had a question for Deepika. Yes.
Yeah, this is actually really inspiring. It made me look into another field when I was like sit on one topic.
I'm going to love it if you do something, Arjun. So, I'm so happy to see your enthusiasm, and man, you had all the right answers. So kudos to you. You are one smart cookie. I already know that. So
all the YouTube time is finally working, Deepika. But I was going to say, over the years, I've heard Deepika say, correct me if I'm wrong, I have to go take care of the mice. I have to go feed my cells. This would always be like the evening job. She would come back home, cook dinner for the family, and then go back. And that's one more thing I wanted to point out. She's an amazing mom, an amazing wife, an amazing daughter-in-law, daughter. I've seen her in every possible role. And for a woman to be able to do all these things so well and have a phenomenal career and do service to the community, you probably get very little sleep, Deepika, but this is amazing.
No, I do sleep very well, and I think you are like the most energizer bunny that I've ever known in my whole life. I used to call her by that name. I said, man, you are like, you go. So you are there very much a part of all our journey. And I think the support system, I can't thank enough how much of a support system the families we know in Madison are like our real families. They are the true families. We have seen our children being born here. Our children have grown together. our husbands connect and argue on so many different things, and we celebrate every occasion. So I think it's so intricate and so close. It's a very close network, and we are blessed to be a part of it too. So yeah.
And Shruti, great job doing a lot of research, talking to Dr. Deepika Rajesh, and being so enthusiastic. We hope to have you in many more such sessions, because I think it'll also be beneficial for you and for us to have a very energetic youngster who can do this very well. You were very articulate too, and it was great to have you. And Deepika, you were very articulate. It was such a pleasure to listen to you. We hope we can do more sessions like this in the future. Just one last word about SciArt Services. I didn't say anything at the beginning because I wanted to, pass on the stage to the speaker itself. But SciArt Services is a non-profit. We're, headquartered in Wisconsin. We're doing work in trying to fill in the gaps in, whatever way we can in STEAMM. And we say STEAMM with a double M here, thanks to Deepika's suggestion. The M is an extra emphasis on music as well. Our motive is to try and encourage a lot of underprivileged students with scholarships as well. In addition to doing a lot of science and arts programming, we're trying to bring a lot of eminent musicians as well. And there is a very nice program called Printable Programming. It's a programming platform called CAD.js, which is on the cloud. And Suresh has conceptualized this for students to be able to learn programming very easily and enjoy it in a very tangible way, using geometric modeling with just JavaScript. And a lot of kids are taking to it. And we have high school students in several parts of the country, in California and Minnesota and in Wisconsin, teaching this to middle schoolers to encourage them to take up, programming and math in, STEM areas as such. And we're looking forward to seeing many minorities and girls take this up as well, because there's a huge gap there. So that's a little bit about SciArt Services. If you have any questions, please, reach out to us. And if you would like to also present a talk on any subject, we would love to have you. So do reach out to us.
Thank you very much, Shruti, and thank you very much, Vanita and Suresh, for this opportunity. And thank you all for joining us on this Saturday morning. Thank you.
Thank you.
Thank you.
Bye.
Thank
Thanks.
you so
Bye.
much. I think we all had a huge takeaway from this, and thank you so much for joining this session despite your busy schedule. And thank you so much to SciArt Services for this wonderful opportunity.
Absolutely. Thank you, Shruti. Thank you, Deepika.
Bye.
Thank you.
Bye-bye.
Thanks, Amol.
Thank you.
Bye.